The School of Biological Sciences at the University of East Anglia is a wonderful place to study as a student, a great place to work as a member of staff, and a fascinating place to visit as a member of the public. We look at how biological systems work by joining together our understanding of genetic information with research on microbes, plants and animals. This includes studies on animal populations and behaviour, plant disease resistance, human health and how microorganisms contribute to our life and environment. It is really important for us to show what we do and to inspire a new generation of scientists. What has the fruit fly ever done for us? We are familiar with these tiny flies gathering round our discarded fruit, but the fruit fly was chosen as an early model for genetics and was the first complex creature to have its genome sequenced. The fruit fly is also useful in the study of development because many of the genes that lay out the body plan in flies can also be found in mammals and even man. We study evolution and mate choice, trying to find out what makes some individuals more successful than others. Mate choice and the effect of numbers of males and females is also looked at using red flower beetles. This activity showed something of the extraordinary value of bees and other small creatures to many areas of biology. Children loved hunting and our lucky dip and spent some time making a model maggot or two. In this activity, children got the chance to meet their own cheek cells. Surface cheek cells were gently scraped off and placed on a microscope slide. The cells were stained and looked at under the microscope. The children got to take a picture of their cells home. Cheek cells were also used as the starting point to isolate DNA. Children were surprised and fascinated to find out how easy it was to get their own DNA, and they took it home with them in a necklace as a souvenir of their day. Many things in biology are too small to see with your own eyes, and even with the help of microscopes. Biologists have developed ways of using fluorescent dyes to see tiny structures within cells. This activity let people use fluorescent microscopes to see cells which have been stained with these dyes. This exhibit showed nests of live ants and a nest of live bumblebees, which was connected to a flight chamber in which the bees could visit flowers. Our research explores why animals show social behaviour, such as living together in a nest, and in the case of bees, to find out more about the basic ecology of these important pollinators to try and protect them. We wanted to show our visitors how bees and ants are kept for study, and how fascinating it is to watch their behaviour, and also how bees play an essential role as pollinators of wildflowers and crops. Microbes have different shapes and can do many different things. For example, the soil bacterium Streptomyces produces antibiotics which we use to fight infections. In this activity, children evolved their own microbe. They gave it a name and decided where it was going to live and what it was going to do. A nature trail around an area of wood at UEA was used to show that even in a small area there can be much diversity in species of trees, plants and fungi. This activity showed how the hugely abundant sulfur molecule dimethylsulfonopropionate, or DMSP, which is produced by many seaweeds and microscopic marine plants, is converted by bacteria to dimethyl sulfide, or DMS. DMS is a gas which gives us the characteristic seaside smell. It is a major part of the marine sulfur cycle. It plays a role in climate change and can be sensed by marine animals. In this activity, seaweed was ground up, and when bacteria were added, the DMS could be smelt. Our eyes are amazing, and in this activity, children were able to learn more about how they work and why our eyes can sometimes go wrong. The airy silk moths come from China. All caterpillars from this family spin silk cocoons, but only a few species are bred commercially for the textile industry. Airy silk production does not involve killing the insects, as the cocoons can be harvested after the adult moth has emerged. Children loved holding these amazing creatures.
This larva comes from a female that was alive last year, so the larva is about a year old.
This activity provided an opportunity for people to build up their own DNA models. After a quick grounding in the basic components that make up DNA and some rules about the construction, people were amazed to find the assembly process was elegantly simple. Children had a go at loading samples into a DNA electrophoresis gel and learnt how an electric current could separate DNA molecules of different sizes. Children came face to face with parasites, including head lice, bed bugs and tapeworms. Learning about the structure of proteins is really important for the design of new drugs to treat disease. Chick and frog embryos are used to study how vertebrates develop. This can help our understanding of both normal and unusual development. This display demonstrated the ability to use bacteria to produce indigo and other related products, which are important as dyes and in the pharmaceutical industry. This is an example of how, by using bacteria as the chemists, these chemical reactions can be done without producing the toxic byproducts that human chemists produce. This activity was Top Gear Biology, human physiology at full speed. It was a fun way for people to challenge their reflexes and ultimately as a way to introduce the physiological effects and benefits of exercise for health and in the prevention of disease. In the evolution activity, children could look at different plants and animals and try to place them along an evolutionary tree.